I'm Naoki Osato at Osaka University in Japan. The title of my talk is Discovery of Directional Chromatin-associated Regulatory Motif Affecting Human Gene Transcription. Double-stranded DNA encoding genes are unfolded where a gene is transcribed in the cell. Transcription factors and cofactors bind to promoters and enhancers and form a transcription initiation complex to regulate the transcription of a gene. CTCF is involved in chromatin interaction. CTCF binding sites are located at the anchors of chromatin interactions. The CTCF binding sites are predominantly oriented in a convergent manner, forward-reverse orientation. Other TFs such as YY1 and GNF143 are also known to be involved in chromatin interaction and insulator function. MIR transposon was reported to be involved in insulator function. CRISPR technology was used to examine the directionality of CTCF binding site. Enhancer-promoter association rules were proposed in a previous study to assign putative transcriptional target gene of a transcription factor computationally. I limited the enhancer-promoter association domains within CTCF binding site and found that the expression level of putative transcriptional target gene was changed according to enhancer-promoter association domains. This suggests that the difference in the expression level reflects the accuracy of prediction of transcriptional target gene using a given enhancer-promoter association domain. The expression level of putative transcriptional target gene was changed according to enhancer-promoter association domains limited within DNA motif site of a putative insulator transcription factor. The DNA motif site associated with a high expression level were coexisted with enhancer-associated histone modification mark H3K27 acetyl, implying that the DNA motif site work as enhancers. I identified 96 biased orientation of motif of TFs that significantly affected the expression level of putative transcriptional target gene in 4 cell types in common. The 96 motifs included non-motif associated with chromatin interactions. The 96 motifs included many motifs involved in chromatin interaction and chromatin-associated functions, such as chromatin remodeling, chromatin accessibility, pioneer factor, and histone modification. A notable example is KLF4, which is associated with three-dimensional enhancer rewiring and transcriptional changes during the reprogramming of mouse embryonic fibroblasts to pluripotent stem cells. The transcription factor USF1 binds within the insulator at a site important in adjacent nucleosomes for generating histone modification associated with active chromatin and by inference with barrier function. About half of the 96 TFs seem to be novel candidates of TF with chromatin-associated function. I examined the co-location of biased orientation of DNA motif sequences and found that some of the DF motif sequences were co-located or overlapped in the genome. I compared the enhancer-promoter interaction predicted using biased orientation of DNA motif site with chromatin interaction data such as high chip and high C data, and found that the putative enhancer-promoter interactions were overlapped more with chromatin interaction data when considering the orientation of the DNA motif site. It is known that two genes closely located in the genome tend to show the correlation of expression level of the genes among tissues and cell types. The correlation of expression level was reduced by biased orientation of DNA motif site, which act as insulator site, such as CTCF. 68% of biased orientation of DNA motifs showed reduced correlation of the expression level. This is a summary of the research. It is difficult to examine TFs associated with chromatin interaction and chromatin-associated function in a comprehensive manner only by experimental analysis. This research will contribute to find such TFs and reveal novel function of the TFs. Thank you for your attention.